Now that you know all the steps of protein synthesis, now we can start doing some of the fun stuff and we can start playing around with the knowledge that you have. So to start this lecture, I'm going to ask you to do a little exercise. And the first thing I want you to do, I'm going to give you an amino acid sequence and we're going to work backwards. But then we're going to mess with the sequence and see what we can do to change it. Say I gave you the following amino acid sequence. Start out with MET, so methionine, because you know that's your start codon. Then you're going to have phenolphthalein. Then do tryptophan, asparagine, isoleucine, and then we'll have a stop sequence. If I were to give you this, in order to do anything unique with it, you've got to back it up. So we're going to work backwards. So with your genetic pinwheel, if you like the one that I always use on page 11, go back to that. And I want you to come up with an mRNA sequence for this. I'm going to give you the one that I came up with, and some of them you don't have a choice, but if yours doesn't look the same as mine, no big deal. So I know if I have methionine, my mRNA code has to be AUG. If I go and look at phenolphthalein, I have two choices. I can pick the one that I chose, because it's simple. There is UUU, or you could also pick UUC. Either one would code for phenolphthalein and you're good. Tryptophan, you've got to use UGG because that's the only one. For asparagine, you have two options. You can do AAC or AAU. And then I'm going to choose the AAC one. For isoleucine, you can choose AUA, AUC, or AUU. I'm just going to take AUC because I haven't used very many C's in here. And then for your stop codon, you've got three options. I'm just going to use UGA, just because that's the one that always comes into my head first. If you want to use UAA or UAG, knock yourself out. You pick which one you want. Now take it all the way back to the very beginning, to my DNA sequence. So T, A, C, and then A, C, C, T, T, G, T, A, G, and then A, C, T. So you can work backwards now. So you better know how to go forwards as well as to go backwards. Now that I've got you to this point, the next thing I want you to do is I want you to cause a mutation. And where I'm going to ask you to cause a mutation is the fourth base in your DNA sequence. And I want you to take it out. So 1, 2, 3, 4. This base is no longer there. And I want you to now go back through and do everything again. Write your new DNA code. I'd still have TAC. I still have AAA, even though I took out that one A. But now, if you'll notice, it messes everything up afterwards. So TAC, AAA, CCT, TGT, AGA, and then I have not quite a codon on the end. It kind of just stops from what I gave you to work with originally. Now work forwards, and I want you to take it into your mRNA code. So AUG, UUU, GGA, ACA, UCU, and then your little stragglers down here, GA. Now take me into my amino acid sequence. I know AUG has to be methionine. UUU hasn't changed. I know that's still phenolphthalein. Now I have a difference though. GGA corresponds to glycine, not the tryptophan that should be there. Then you have ACA, which corresponds to threonine. Then you have UCU, which is serine. And technically, you would have a stop here because you can't go any further. But you wouldn't necessarily have to put that. You can just kind of assume that you're going to stop because you don't have another codon. So if you compare your two amino acid sequences, your two proteins, what's different about them? The methionine is the same. The phenolphthalein is the same. But after that, I have a completely different protein. When you do that, then you've probably messed something up big time. Because glycine is totally different than tryptophan. Threonine is totally different than asparagine. And then isoleucine is totally different than serine. One thing about proteins when they're in your body, once a protein is made, proteins always fold in a very distinctive way. Each protein has its own unique fold. And if you want to see this, I can definitely show it to you in class. When I was doing research in college, I looked a lot at proteins and how they fold, because if they fold wrong, they can no longer do their job. So now that you've made a change and I have a different protein from here on, my protein's not going to be able to fold and it's not going to do the job that it has to do. So, what do you think that you just created? Hopefully you're telling me you just did a mutation. And that's what I will focus on for the rest of this lecture. A single deletion can cause a mutation and can cause a protein to no longer work. We talked about these when we were doing chromosomes, and I talked about all those different chromosomal mutations, and we talked about the different disorders that come from that, like Cru de Chat, and we talked about Down syndrome, and we talked about Turner syndrome and Klinefelter syndrome, and a couple of other ones. That's when something happens to your chromosome and you have those inversions, the deletions, additions, all those different things. These mutations are different because now I'm talking about the root of what makes you you, and that's your DNA. I caused a boo-boo in your DNA, and then that has a domino effect throughout the whole process. If you get a mutation in your DNA, it's going to get replicated with that mutation, and that can cause a lot of problems. Then when you go into our new process of protein synthesis, you could totally screw up the protein that needs to be made, and you're not going to be able to do what you need to do. One of the things that I will talk about in terms of mutation at the DNA level will be sickle cell disease, and we'll talk about that when we get into class. Now, with mutations, there are two major types of mutations. One of them we've already talked about, and that is the chromosomal mutations. And if you'll remember back to those, you're talking about additions, deletions, inversions, substitutions, those types of things. The ones that I'm going to talk about now that specifically kind of have to do at the molecular level, the DNA level, those are given the term point mutations. And these are the ones that I will focus on mostly with DNA. For a point mutation, kind of your broad definition is a change in one of the base pairs of DNA. It could be just one change or it could be a couple of changes. It just depends on what actually happens to it. So it doesn't have to be just one. It could be one or more. But a lot of times a point mutation is all you need to make a drastic change in the protein that you need to make. Before I continue on with this, what do you think causes point mutations? What is the biggest cause of them? What actually causes it? We call them a mutagen. A mutagen simply is something that causes a mutation. Examples of a mutagen, you can have chemicals. If you're exposed to a certain chemical over a long period of time, it could cause a change in your DNA code. For example, off the top of my head, a long time ago, and this is only probably about 12 years, you guys probably think it's forever ago, but for me it's not that long. When I was a senior in high school, I believe this is when they made the change. All specimens that we had for biology and what I used for anatomy, or if I had been teaching back then, would have been kept in formaldehyde. And they actually found out that formaldehyde is a carcinogen, so it's no longer allowed in schools. So my cats that I have for anatomy, that wonderful smell that you guys keep smelling now, isn't formaldehyde anymore because we can't use that. So it's a different chemical. But if I had maybe been an anatomy teacher and had been teaching for 20 years, that continued exposure to formaldehyde could have caused a mutation in a cell, causing me to have cancer. So an extended exposure to a chemical could cause a point mutation in your DNA. One other thing that you guys should be very familiar with, radiation can cause point mutations. And then you can also have just simply replication errors. And we will talk about that in an exercise that we'll do in class. So these are just some examples of where you might get it. Chemicals, radiation, and then sometimes mistakes happen. And when you go from tryptophan to glycine and you completely change what amino acid gets brought, then you're talking about a boo-boo and a pretty serious one. With point mutations, there are a couple different kinds. I'm going to ask you to figure this out, and this is what I want you to bring into class. And I'm going to do it with numbers that actually spell something. And I want you to try to come up with the names for these different types of point mutations. And don't make it hard. Keep it really simple and tell me what's going on as I go through the different examples that I'm going to give you. So to start with, this is your original protein. The cat ate the rat. That's your original. Everything else is going to be some variation of that, and I want you to come up with what you think its name would be. Your first one is the CCT ate the rat. Here's my boo-boo. Your second one that I want you to take a look at, the cat, TET, her at. And then the last one that I want you to figure out and use to see if you can figure out the name, the cat, eat, era, and then T all by itself. So looking at the different boo-boos, and here's kind of your boo-boo there. The boo-boos on this, I want you to come up with the names for these. Now keep it simple. Do not make this harder than what it is. Bring this information to class, and I will leave it here.